Yeah, sure. Thanks, Gabriel. I'm Lexi Davis. I'm a Great Lakes Water Resources Scientist with the Great Lakes Program.
So the trained user networks in the Finger Lakes originated before NYHABs began, and they're aimed towards local groups to monitor for harmful algal blooms at routine locations of interest. Trained users monitor specific locations routinely, and zones are pre-established by coordinators. We'll go over zones a little bit more further along in the presentation and how those exactly those are coordinated between us and your individual surveillance programs. With the inception of NYHABs, trained user groups continued to operate in similar manner with the addition of online reporting to DEC through the NYHABs program. And trained user groups empower and educate local communities to monitor and educate around harmful algal blooms. So a rough summary of what we'll go over tonight. We'll do a summary of the 2025 NYHABs report, just to kind of help you all visualize how and when HABs reporting is happening in the Finger Lakes last year specifically, to make things a little more interactive and visual. We'll do a harmful algal bloom introduction and overview. We'll go over New York State DEC's holistic approach to harmful algal blooms. And then finally, we'll do the training around harmful algal bloom identification and the NYHABs platform. So with that, I'll hand it over to Lexi.
Okay, so
in advance.
Thanks, Gabriel. I will kick things off by going over some of the 2025 NYHABs season data. I pulled this from the Division of Water Monitoring data portal and summarized all the data reported to NYHABs in the Finger Lakes region. Go ahead. Okay, so on the left is a map showing reports submitted to NYHABs for each of the Finger Lakes in 2025. The colors indicate the harmful algal bloom or HAB extent, with widespread blooms being dark blue, large localized in dark green, small localized in light green, and open water in blue. The no bloom reports, which some of you provided, are also included, and those are indicated in white. So in total for the Finger Lakes in 2025, there were 837 confirmed HAB reports across 10 Finger Lakes. Canadice Lake did not have any confirmed harmful algal bloom reports in 2025. And there were 965 no bloom reports, mostly on Keuka Lake. And there were 153 total days with confirmed HAB reports. The chart on the bottom shows the breakdown of the HAB extent for the confirmed reports. So most of the HABs were characterized as small localized blooms or large localized blooms, with some widespread and open water HABs, but far less than the other types. We also summarized the HABs, the confirmed HAB reports by date, to display the length of the HAB season. So this figure shows the confirmed HAB report dates in green and no bloom reports in black. So generally, the first occurrence of a confirmed HAB was in late June to mid-July, but there were a couple earlier observations of confirmed HABs in some lakes, such as Keuka and Canisius Lakes in 2025, who had blooms in early June. And the last occurrence of a confirmed HAB was through October, but Honeoye notably reported confirmed HABs into later November. So looking at the chart where most of the green points or confirmed HAB reports cluster together are generally in mid-August to early October for most of the lakes. We also wanted to share that we broke down the reports for each lake as well. So for Canisius Lake up first, there were 7 confirmed harmful algal bloom reports and no bloom reports, with the first occurrence of a confirmed HAB being on June 12th and the last occurrence being on October 26th. Most of the reports, well, about a 50-50 split, were from the Department of Health and the public, and most of the blooms were categorized as small localized blooms with some large localized blooms as well. For hemlock and Canadice Lake, like I mentioned before, Canadice Lake did not have any reported confirmed blooms for the 2025 season. But for hemlock, there were 2 confirmed harmful algal bloom reports and 66 no bloom reports. The first occurrence of a confirmed HAB was on July 28th, and the last was on August 12th. All of these reports were from the Rochester Water Board, and like I said, most were no blooms, but the rest were small localized blooms. For honeoy, there were 70 confirmed harmful algal bloom reports and 2 no bloom reports. The first occurrence was on June 30th, and the last was on November 21st. There was a good portion of the reports submitted by the public, but there was a good portion submitted from Honeoy Lake as well, with some others from agencies like DEC, Parks, and DOH as well. And most of these were large localized blooms, but there were some widespread and small localized blooms mixed in there as well. For Canandaigua, there were 74 confirmed harmful algal bloom reports and no bloom reports. The first occurrence was on July 28th, and the last was on November 3rd. Most of these reports were through the Canandaigua Lake Watershed Association, and there was about a 50-50 split between small localized and large localized blooms. For Keuka Lake, there were 53 confirmed harmful algal bloom reports and 521 no bloom reports. The first occurrence of a confirmed HAB was on June 8th, and the last was on November 27th. Most of these reports were submitted through the Keuka Lake Association, and like mentioned before, most of these were no bloom reports, but others were small localized and large localized blooms. For Seneca Lake, there were 159 confirmed harmful algal bloom reports and 1 no bloom report, with the first being on July 22nd for the first confirmed HAB, and the last being on October 28th. Most of these were from Seneca Lake Pure Waters Association and a good portion from the public as well. There was about a 50-50 split between small localized blooms and large localized blooms, with some widespread in there as well. For Cayuga Lake, there were 285 confirmed harmful algal bloom reports and 24 no bloom reports. The first confirmed harmful algal bloom in 2025 was on July 8th, and the last was on November 5th. Most of these were reported through the Community Science Institute and some through the public and parks as well. Most of these were small localized or large localized blooms, but there were some widespread as well. For Owasco, there were 102 confirmed harmful algal bloom reports and 161 no bloom reports. The first confirmed HAB was on July 28th, and the last was on October 28th. And these were mostly through the Owasco Lake Watershed Association, with about a quarter of them from the public. Most of the reports were no blooms, but there were some small localized and large localized blooms as well. For Skinny there were 51 confirmed harmful algal bloom reports and 84 no bloom reports. The first occurrence of a HAB on Skinny Atlas last year was on August 22nd, and the last was on September 30th. Most of these were submitted through the Skinny Atlas Lake Association, but some from the public as well. Most were no bloom reports, but the rest were generally small localized or large localized blooms. And last but not least, Otisco Lake had 45 confirmed harmful algal bloom reports and 107 no bloom reports. The first confirmed HAB was on June 28th, excuse me, and the last was on September 16th. Most of these were through the Otisco Lake Preservation Association, and most of the confirmed blooms were either small localized or large localized, but the majority of the reports were no bloom reports. Okay, so now that we've summarized the 2025 NYHABS season, we'll go into what are harmful algal blooms. So this will be a refresher for some folks, but still good to have some reminders. So we're going to start with what algae is, or what algae are. So algae are single-celled protists that have chlorophyll, which provides that green color, and they photosynthesize. So they use sunlight to make food from carbon dioxide and water, and they are normally not harmful and are naturally occurring. And I'm sure you are all well aware, but the acronym HAB stands for Harmful Algal Bloom, and despite algal in the name, a HAB actually refers to cyanobacteria that are not true algae. So cyanobacteria are also natural, and they're very specialized and competitive. Ancient bacteria that have been present for billions of years, and they're able to do things like regulate buoyancy or their ability to float, or fix nitrogen in order to fill specific niches. And they also have other pigments in addition to chlorophyll, so they can present in colors other than green, which is why you may have heard them also be referred to blue-green algae. And they are often hard to predict and difficult to remove, as we are still trying to learn and understand as much as we can about them. And under the right conditions, these can form harmful algal blooms, which produce toxins, which we'll take a look at on the next slide. So there are lots of different toxins that can be produced, but I'm going to focus on just a few. So the first is microcystins, which are liver toxins, and they're produced by cyanobacteria such as Microcystis. And this is what is most commonly observed in New York. The next is anatoxins, which are nerve toxins, and they are produced by cyanobacteria such as Delicospermum, which is also common in New York. This one is specifically known for its ability to fix nitrogen, where it converts nitrogen to a different form, such as ammonia, using a catalyst. And now, while some cyanobacteria produce microcystin and anatoxins, some of the time, all cyanobacteria have lipopolysaccharides, which are endotoxins that are skin irritants and allergens. So all of that to say, there are no visual cues that toxins are present or what toxin is present. So there can be a bloom that is visually intense, but toxins may not be present. Or there may be a bloom where it doesn't look that bad, but the toxins are present. So this is why it's really important to know what HABs look like, avoid it, and report it when you see anything that looks like a HAB. So we can be exposed to these toxins in many different ways, through consumption by accidental swallowing or directly drinking the water, inhalation through aerosols being created during household use or recreation, or dermally through skin contact during swimming. And I want to emphasize that any of these health effects should be reported through your local health department if you or someone you know has experienced them. So symptoms of HAB reactions include allergic or irritation to the skin, eyes, ears, or throat. There can also be some gastrointestinal issues such as nausea or vomiting. You can have difficulty breathing, discoloration of the skin, or jaundice, or neurological symptoms like dizziness, confusion, and headaches. And like I said, again, in the last slide, make sure to report if you're experiencing any of these to the health department. So a common scenario where we see HABs is when there are high temperatures, high light, and high nutrients. But as we all know, HABs have been present in low nutrient water bodies too. So historically, HABs have been associated with high nutrients, with total phosphorus being greater than 30 micrograms per liter. But HABs and high nutrients are not always neatly linked, as evident by HABs present in low nutrient lakes, much like the Finger Lakes, with some having average total phosphorus values much lower than 30. So therefore, bloom triggers are not fully understood, and there are some low phosphorus systems that bloom, but some high phosphorus systems that don't. There's also interannual and seasonal variability within lakes that can impact HAB bloom triggers. So for seasonal variability, it's important to look broadly at the ecosystem picture, as cyanobacteria aren't the only things in the lake throughout the year. In the figure, we see the abundance of phytoplankton throughout most months, but you'll notice peaks of different species throughout the year. So diatoms dominate in the spring and fall, which leaves an opening in the summer for the blue-green algae to be dominant. Which makes sense in when we really see the HABs on the Finger Lakes. But it's important to note that the blooms have been documented in New York State from April through January, and there are other factors that continue to widen the window of when we expect blooms to occur. So research has shown that there are several parameters associated with the presence of harmful algal blooms. So phosphorus, dry-sended mussel presence, lake fetch, which is the length of the lake surface over the wind can blow uninterrupted for a single direction, and compass orientation of max fetch, where these are all significant parameters closely associated with the presence of HABs. So due to their north-south orientation, the Finger Lakes specifically check off the last two boxes of those described. And all of those things coupled with sunlight, calm conditions, and warm temperatures makes for perfect conditions for HABs to occur. But again, it's important to note that some model parameters cannot be managed, and phosphorus is specifically called out as it is currently the only factor we do have some control over. And of course, there are some wild cards affecting HABs that are out of our control. So with climate change, there are warmer temperatures and longer periods of lake thermal stratification and episodic nutrient delivery from intense rain events, followed by long periods of calm, stagnant conditions, which can really be a trigger for HABs. There's also select feeding by dry-sended mussels that contribute to HABs, with research out of the Great Lakes showing that mussels can intake and actively reject microcystis and select for the better green algae and diatoms. And emerging contaminants could also play a role in bloom triggering in ways that we aren't sure yet. I'll turn it over to Gabriel next to discuss DEC's holistic approach to HABs. You're muted, Gabriel.
Thanks, Lexi. I'll quickly go over DEC's holistic approach to HABs and some of the planning and other mitigation efforts that DEC undertakes. So DEC recently released its HAB roadmap, and New York State has a long-term strategy with a holistic and integrated approach to harmful algal blooms with several pieces in focus. The first is watershed management through monitoring, clean water planning, and pollutant reduction strategies to address long-term controllable causes of HABs. The next is funding projects to reduce the occurrence of harmful algal blooms through programs like the Water Quality Improvement Program and non-point source planning grants and other funding sources. Next is research on HABs and their causes with a focus on prevention, mitigation, monitoring, and modeling to understand causes. We have available the New York State DEC HAB research guide that highlights specific areas of interest in HAB research. And lastly is in-water mitigation through development, implementation, and evaluation to help control bloom size and formation. And the HAB roadmap is supposed to strategically lay out and organize those different priorities for addressing HABs in New York State. There are 6 focus areas covering important aspects of managing HABs. Each focus area includes a goal statement and associated projects and tasks to improve the understanding of HAB drivers, reduce impacts to health and recreation, and to take action both in the short term, the next 1 to 5 years, and the longer term, 5 years and beyond. Specifically, those focus areas in the HAB roadmap include monitoring and assessment, water quality standards and regulatory thresholds, planning and implementation, general in-water body mitigation research, and permitting in-water body mitigation practices, and finally, public outreach and reporting. All right, thank you, Lexi.
Okay, thank you. So next, we will go through and highlight a few pieces of HABs research, which we use to inform management. So I want to first speak on a piece of research completed by the state that uses the HABs analytical data, beach closures, and public reporting from 2012 through 2020. So this work was completed by Rebecca Gorney and the team, and they analyzed 24,000 records on 655 water bodies for frequency, or the reports of HABs across New York State water bodies, intensity, blue-green algae, and chlorophyll to understand potential changes over time, as well as duration, looking at the average beach closure. So they found that for frequency, half of the lakes tested had only 1 year in which a HAB was documented, and less than 1%, or 6 lakes, had a HAB documented every year of the study, which is shown in the bar chart on the side. For intensity, there was a significant increase in the number of HABs reports each year, but not an overall increase in intensity, based on evaluation of chlorophyll levels in mid-lake samples collected routinely. The increase in the number of HAB reports can be attributed to larger awareness or more people reporting. And for the duration, it did not lengthen over the study period based on beach closure data captured. So next, I'll go over the HABs Advanced Monitoring Pilot. So the project was a collaborative effort between DEC and USGS staff and included 4 main strategies to achieve the project's goals, including tributary monitoring and assessment, automated monitoring platforms, nearshore mapping, and intensive lake characterization. So as a result of this effort, there were 6 data releases providing all data collected from the pilot, including in-water body sensor data, other environmental conditions, lab, biological, and nutrient data, algae and cyanobacteria counts and identification, and data on stream health, habitat, and hydrology. And that's all available on the DEC website. Another piece of work that is currently under review is striving to alleviate the observer bias through the use of satellite imagery to provide a more broad and unbiased observation of HAB occurrence statewide by using the satellite Sentinel-2, which offers a 5-day flyover at high 10-meter resolution, allowing application to relatively small lakes across the state, like the Finger Lakes. So this takes away the observation bias introduced through targeted monitoring, such as what we have with NYHABs, where we often receive multiple reports on the same day of the same HAB. So this uses Google Earth Engine, and we're able to estimate concentrations of phycocyanin, which is a blue-green pigment found in certain microalgae and cyanobacteria. So here's an example of the application of that method on Seneca Lake in 2024. So on the left, we have a poll from NYHABs showing the confirmed reports and an image from one of those reports. And in the middle, we have a satellite image of the northern end of Seneca Lake showing a pretty widespread HAB and an image taken by plane of the event as well. And then on the right, we have the outputs of the satellite work, which shows the estimated concentrations of phycocyanin in micrograms per liter. And we hope that this work can serve as a way to reduce bias and evaluate HABs before and after implementation of management actions that may be included in things like clean water plans.
Awesome. Thanks, Lexi. So I know a lot of that was kind of higher level information that some of you might have come here tonight expecting. Some of you might be more interested in the identifying HAB portion of this, but we think it's important to give you a holistic idea of what DC is doing to research and combat HABs, some of the history of harmful algal blooms in the Finger Lakes, and how some of the data that you all help collect can show up on our publicly on the data portal and be used by ourselves and others post the monitoring season. So now we'll jump into the training portion and of HAB identification and the NYHABS platform. So first we'll play our usual to HAB or not to HAB game. First we'll do a little identification and then we'll test your skills afterwards. So first we'll go through some examples, some common examples of things that are often mistaken for harmful algal blooms, but are not. Filamentous algae on top, it often appears like wet cloth or hair. It's harmless, and often that we use the stick method sometimes. If you can put a stick in it and it lift it out of the water and it looks like hair or wet cloth, not a harmful algal bloom. The middle picture is duckweed, very small plants. You can see the individual little plants in that close-up picture, and that also is usually pretty easily visible if you can get close to the suspected harmful algal bloom. On the bottom is pollen, usually in spring and early summer. It's very yellow and it breaks apart. The breaking apart can be an easy way to differentiate because sometimes it can look very close to a harmful algal bloom. This is more filamentous green algae. So we like to start with identification by telling people you're able to tell how you're able to tell is not a HAB. And Cladophora is often one of the most misidentified algae. This is filamentous algae that can accumulate on the top of lakes and can look like a HAB. The growth starts on hard substrates. As photosynthesis really ramps up, air bubbles accumulate in the filaments, and the Cladophora is broken free to accumulate in mats that get pushed up, or pushed up and around, similar to harmful algal blooms. And this is very common that we get Cladophora reports misidentified as HABs, and there are a couple of other types of filamentous green algae. This is Spirogyra. It's bright green. It can look like a cloud when it's in the water. When you take it out of the water, it doesn't anymore. It contracts and looks again like that hair, wet cloth type of texture. Some more examples of Cladophora. It can often wash up on beaches and mats, coat hard surfaces like the rocks in the top pictures. And at first glance, it can give a similar green color, like in the bottom left, to what the pea soup type of harmful algal bloom might appear like that we'll look at in some of the later pictures. Some key differences between Cladophora and a HAB. Cladophora forms dense mats in water, often on the bottom or on the shore. It's not known to produce toxins. It usually peaks in early summer, and it's filamentous, meaning end-to-end growth. Harmful algal blooms, on the other hand, are floating blooms that tend to stay in the water on the surface, and they can produce toxins, often peak in late summer, but not always, like Lexi went over, and they're colonial. A little bit more on duckweed here. Duckweed is another phenomenon that can resemble a HAB. Duckweed bears seeds and is free-floating. It can grow at remarkable rates, sometimes doubling in population in 48 hours, and it can easily coat the surface of a lake or a pond. Duckweed also has tiny roots. There is also a similar algae called watermeal, pictured on the two fingers, that is tiny, firm, and grainy, and are double ovals in shape. These are also harmless. Watermeal is generally smaller than duckweed and it does not have roots. A lot of times they will grow together in a water body. So pollen is common in the spring, especially near pines, and it will present bright yellow usually. HABs are typically not this shade of yellow or yellow at all generally, and the pollen will break apart when it's touched with a stick or something. And doesn't usually coat a stick if it's pulled through the accumulation. It's most common in the early summer or spring, like I said. It's an example of pollen on Skinny Atlas Lake in June of 2020. Another example from Owasco Lake. So now we'll go over examples of some actual HAB identification. So things that are likely to be harmful algal blooms often appear oily and shiny. They can have a pea soup appearance, especially when mixed in the water column. They can be surface scums or foamy. They can look like spilled paint on a surface, and they can be discolored streaking and present that green, blue-green coloring that we've talked about. And they can also be floating clumps or globs, like in the bottom right there. So here's the oily, shiny characteristic. You can see the very obvious blue-green color and also the streaking from left to right, sideways across the picture there. All three of those characteristics are very characteristic of a harmful algal bloom. This one slightly harder to identify, but this is the pea soup, scum, or foamy characteristic that we talked about. This isn't the best picture, but you can see that the water is very discolored and the color of pea soup very green, and this bloom looks like it's mixed more through the water column, and then further out, it looks like there's some scum and foaminess and also some streaking if you look really closely, even though there's a glare. Again, with the paint, scum, or foaminess, both of these, for me, the most identifiable characteristic is the streaking. You generally don't see that in some of the other types of algae, pollen, etc. that we were talking about. It really does look like a paint oil sheen almost on top of the water in both of those pictures. So HABs can also be localized on the surface or extend down into the water column. Some species of HABs are able to regulate their buoyancy and make vertical migrations according to light availability. This is a good example of a HAB that's throughout the water column. This one's a good example of one that's on the surface, but also streaky and could be below the surface into the water column. HABs often accumulate on shorelines, docks, or coves. This is often from them being wind-driven on the surface, like Lexi talked about earlier with lake fetch, where the direction of wind and the lake orientation can help HABs accumulate in these sheltered areas sometimes. Another example of accumulation on a beach. And this one, you can see the streaking, the mixing through the water column, the pea soup color, foaminess, all characteristics of a harmful algal bloom. This one's slightly less common, I would say. This is some streaking, clumps, or globs, and HABs can sometimes present in clumps or collections of cells, like in these two pictures. So now we'll test your skills and see if you guys think these are HABs or not HABs. So first, the one on the left, and feel free to unmute yourself and take a guess or guess in the chat. There aren't really that many people on the call, so everyone can participate if you want. Otherwise, it'll just be me talking. So the one on the top left, does anyone want to guess if that's a HAB or not a HAB? So it's pea soup color. That's the most easily identifiable characteristic for me in that picture. How about the middle top with the Secchi disc? That one's a little harder, but we would say that that's not a HAB. It just looks like slightly turbid water, so some sediment mixed in the water column possibly. There's no green-blue color, no foam, streaks, clumps, anything like that. Nothing to indicate that it's a HAB. Yeah, other than some turbidity, some sediment cloudiness in the water. The top right, any guesses?
Yes.
Yeah, that's an easy one. So yeah, blue-green streakiness. Yeah, I'd say those are the top characteristics. How about the middle left?
Probably.
That one
No.
wouldn't be a HAB. That's some filamentous green algae. And it looks like the water's pretty clear above and below. And then the algae in the middle, if you put a stick or something like that into it and pulled up, you'd see the filamentous green algae. Oh, skipped that one. The middle one would also be a HAB. You can see the very apparent streaking, like the spilled paint color that we talked about. Some little specks of clumps of collections of cells as well. How about the middle right?
Yes.
Yeah, good guess. That one's hard to tell from the picture. It's kind of similar to the top middle one, other than more green. And from that picture, it's hard to identify exactly if there is a harmful algal bloom. And we can, when we go over picture submissions in later slides, this will be a good example of if there is doubt always, but especially if there's doubt over whether it's a harmful algal bloom to submit really good pictures to try to discern. Bottom left.
Yes.
Yeah, it looks most likely like a harmful algal bloom. That picture also is pretty blurry and would be much more easy to identify if it were a better picture, but you can see streaking and those clumps like we were talking about before. Bottom middle.
No.
Yeah, probably not. There's lily pads, and then looks like filamentous green algae in the top middle. If the picture were a little closer, you could see into the water column a little bit, but yeah, most likely not. How about the bottom right? Yep, that one's not. More filamentous green algae there. You can clearly see the plant matter. All right, thanks for playing. So now we'll go over the NYHABS platform, the New York Harmful Algal Bloom System. So the New York State HABs program is an interagency collaborative effort between DEC, the Office of Parks, Recreation, and Historic Preservation, and the Department of Health. DEC coordinates several HABs reporting efforts, and DEC operates NYHABS to provide a central resource for communicating harmful algal bloom occurrence statewide. And that public awareness is the chief use of NYHABS, among others. And I'll talk a little bit more about why that is important. Also, it's used to convey health concerns and drinking water treatment. So harmful algal bloom reports, NYHABS reports, are conveyed to local water system operators and the Department of Health. The Department of Health evaluates NYHABS reports for potential impacts to public water supplies. And all Finger Lakes except Honeoye have public water supply. Timely report submission is important for that reason, for the Department of Health to be able to make public water supply risk determinations. If reports are submitted days, especially weeks later, they become much less or not at all useful for water supply determination, risk determinations. These reports are also used for regulated swimming areas, beaches, and they serve to help with a protective response protocol based on visual observations for beach closures. Some background. Harmful algal blooms have increased across the world and across the United States in recent decades. Between 2012 and 2025, DEC confirmed reports of freshwater HABs in over 700 water bodies statewide, and in each year HABs are reported in new lakes. In response, the New York Harmful Algal Bloom System, NYHABS, was established in 2019. So why do we operate on a visual-based response? So symptoms are possible with or without toxins. Again, blooms are dynamic. Their spatial, temporal, and toxin gene expression can change very quickly. Sampling and analysis takes time. Those 2 points are directly in conflict. By the time a sample is analyzed and either confirmed to be a HAB or not a HAB, that HAB can have dissipated. Its toxin expression can have changed to be either toxic or non-toxic. And the bloom could have moved, grown, shrank, disappeared, etc. Not all toxins are analyzed, and there's not a practical way to always sample all waters. Like with those maps that Lexi was showing as an easy example, it would be a massive undertaking to try to sample every one of those locations within each water body that were sampled, that were reported, sorry. And so this is DEC's way to be able to most effectively identify and report HABs across the state. An example of a HAB that from that close-up picture looks very evident and present on 16th and 17th. The water is completely clear, and the HAB is either completely dissipated or is elsewhere in the lake. So that brings us to you all, the role of volunteers. What do volunteers need to do? HABs surveillance volunteers agree to survey specific zones on your lake weekly at about the same time. Remember that reports stay live on NYHABs for 2 weeks, so only one report per location is necessary per the reporting guide. It's not necessary to send reports daily for the same HAB event. Submitting a report whether a bloom is present or not, we're asking. So no bloom reports if there is no bloom present, and a confirmed HAB report if you observe a bloom. We ask that you document latitude and longitude coordinates with the radio button. Sarah, can you explain what the radio button looks like better than I can?
Yeah, it's just the map. So like when you go in to click your organization and then your lake or like your water body, it'll automatically populate a little point in the map. And so that's great, but we're trying to make reports more, I don't know, I guess more correct by doing the lat long. So like if you're on the shore of Cayuga Lake and you press Cayuga in your water body, it'll default to like the center of the lake. But if you're on like the southwest shore and you want to make it more correct, you can use that radio button or that map to update your point. So it's essentially just clicking on the map and pointing where you're at to make it the most accurate.
So if a HAB is present, take pictures and submit a bloom report, and 2 photos if possible, 1 close up and 1 further away. And we'll go over some examples of good and bad pictures. And just to reiterate the first part, for public reports, we request only 1 report per 2 weeks because the primary goal of NYHABS is a public reporting, a public awareness tool to make people aware of where a HAB has been observed. And to fulfill that function, we don't want people submitting a report every single day in the same location for the same HAB, because then the NYHABS platform would just show many reports and wouldn't provide additional information to the public to show that there was still a harmful algal bloom there. So we asked for 1 report for 2 weeks, but for the surveillance programs that you all are participating in, you're agreeing to submit 1 report per week. So photos are very, very important. We follow up sometimes with someone if they've submitted pictures that aren't very clear, but to avoid that, we asked for 1 close-up and 1 far-away picture. These are some examples of not great pictures. We asked that pictures not include any people, especially children, that you be close enough to see the water. If there's something like a boat or a dock, that would be helpful for perspective. So the first picture is too far away. We can't really see what's going on in the water, and it includes people. The second picture has too much of a glare. You can't see into the water at all to see what's going on there. The third photo also has too much of a glare, and it's hard to see the perspective of the pictures. There's nothing to kind of gauge off of. And the last photo also has too much glare. Also, please don't use AI or any other tools to edit your images in any way before submitting, because that immediately takes away all of the reliability and usefulness of that report, the data pool that it's contributing to, etc. Also, please don't share pictures. We sometimes have more than 1 person use the same photo because they happen to not get a picture and ask their neighbor for one or something. And all of those things can deteriorate the quality of the report or the data. Some updates for 2026. No major changes, but confirmed bloom will be determined from visual reports, like I said, consistent with the Department of Health protocols to close public beaches and public messaging to avoid blooms based on visual sightings. This improves our reporting efficiency. Again, take photos and submit with no bloom reports also. So photos, whether bloom is present or not, please. So a little bit about zones. DEC is not adding new zones to trained user form due to the way the new database is set up. How zones will be used is primarily at the discretion of your individual volunteer coordinators and programs. And that shouldn't really change much about your interaction with DEC. You all were already working with your program coordinators to work on assigned zones. So that's something that will be a conversation with your coordinator. Zones can be used to coordinate volunteers by individual groups, but the geolocation is the most important data point for us in NYHABs. So geolocation, like Sarah was talking about, the specific coordinates of the report. It's important when selecting a zone that the report will automatically snap to the coordinate of the zone that's loaded into the NYHABS portal. So the reporter must update the location to prevent an inaccurate report location from being submitted. This can be done manually or by using the Find My Location icon. So when you're submitting a report, and you select a zone, please update the location after selecting a zone, because it will automatically snap to a location that is not the actual location that you observed the harmful algal bloom. So reports stay live on NYHABS for 2 weeks. Only one report per week is necessary for trained users like yourselves to avoid excessive reports. So NYHABS is an ArcGIS online interactive map of HAB reports. It's updated Monday through Friday. Reports include status, extent, reported by, exact location, and photos, like Lexi was showing in her slides at the beginning of this presentation. Current reports are active for 2 weeks. No need to report a HAB every day, like I've went over. Archived reports, all previous reports of the year are available on the portal. You just need to turn on the archived report bar, and it will show you older reports from the season. And users can also filter by lake, county, and export reports as PDFs. So NYHABS uses Survey 123. It's an Esri ArcGIS online fillable form. It works on any platform, so you can do that from your desktop, mobile, or tablet. Although, like we were saying with location reporting, it's often easiest to do the report where you observed it, so you don't have to go back later and try to fill in a location accurately. But you can fill it out anywhere. Attach photos to the report, and that's required in order to submit it. And you'll be using the trained user report form. You'll select your user group, which is your watershed group, your HAB surveillance coordination group. This is just a little diagram of how reports go through DC and DOH evaluation before going on the NYHABS portal. And you all submit some additional information that the public form doesn't contain. And we'll be sending out this presentation and the recording, so you'll have these links, so no need to try to scribble them down, but you'll have access to the HAB Pro form again. Some key takeaways: volunteers should use the Pro form to submit bloom reports. The public forum is available for you as an outreach tool as well. Photos will be required to submit a bloom report. We're asking for photos with no bloom reports too. No sampling is involved in these efforts. Please use the radio button to document coordinates for accuracy. Only submit one report per week per location, and reports will stay live for 2 weeks. This isn't to say that if you're in a separate location within that time frame and you see a bloom, absolutely, you can submit a report. This is just specifically pertaining to the location that you're volunteering under your surveillance program to monitor once per week. No major changes to the Pro user or public form. And you can reach out to habsinfo@dec.newyork.gov for questions to DEC specifically about NYHABs and HAB reporting. And that's always monitored by a DEC person, so you'll get an answer pretty quickly. And also, most of the information that we went over tonight, besides specifically the trained user portion of this, is available on DEC's website. You can just Google harmful algal bloom DEC and this contact info, and all of the information about the HABs roadmap and harmful algal bloom program with DEC will come right up. You can also reach out to myself for Finger Lakes related questions, or Lexi, and Lewis McCaffrey is also in the Finger Lakes watershed program with DEC. And again, at the bottom, that HABs info email address. So that's all I have for tonight. But myself, Lexi, and Sarah are happy to answer any questions. I know the reporting frequency and some of the other things can be a little confusing sometimes. So if you have any questions about anything that we went over, feel free to unmute yourself, put in the chat, whatever works.
Thanks, Gabriel. So something I did want to note, I know you mentioned that the NYHABS platform is updated Monday through Friday, but I just wanted to ask if it's okay that people would submit a report on Saturday or Sunday, and what would happen with that report if it were submitted on the weekend.
Yeah. During the HAB season, there's someone monitoring the portal, or there's someone evaluating reports from yourselves Monday through Friday. So reports that come in on Saturday, Sunday will be evaluated on Monday and will go live after that. And also, Sarah, do you want to confirm when NYHABs is going live? Or
That's a great question. So in theory, it's supposed to go live tomorrow, but I think they've got to sign off on it. So we'll see what happens throughout the day tomorrow, but definitely before the first week of June, there will be like a news press release that comes out with DEC. And then if you go to the HABs website, there'll be a blurb at the top in yellow that's like NYHABs is open now, and then you'll be able to actually look at the dashboard, because right now it says it's closed. So the hope is tomorrow, definitely next week, if not tomorrow. And then another thing just about the weekends, the reason we don't look at it on Saturday and Sunday is also because DOH doesn't evaluate public water supplies on the weekends as well. So we don't want to, I don't know, it wouldn't help a whole lot from the public water supply standpoint. But we still, I don't know, I think Brian still looks at it sometimes, but we don't, anything that comes in on Saturday, Sunday will be confirmed first thing Monday morning.
Thanks, Sarah. Robert, do you have a question?
Yeah.
Yeah. My question's about the satellite data, and it's kind of a technical question, but I was curious about two things. One was that you were using the Centennial C, and I've from my research, I've I've seen people use the L2 or like the other Centennial 2 satellite, but also I was curious on the code or the filtering JavaScript that you're using to determine phycocyanin.
Yeah, I can answer that one. So Sentinel-2 is resolvable for smaller lakes across New York State. And so previous research we've done has used that. Sentinel-1, 2, Sentinel A, B, and C have all launched, and they all have a 1 and 2 version of it. So I think, like, if you go into the Copernicus website, it'll say Sentinel-2B 1 and 2. Like, you can pull images from both of those. So there's like a ton of images available. Sentinel, the C launched in like 2024. I don't know if the second one has come out yet, but yeah, there's a lot of images you can get for the same day, but getting a full data set is really hard because of all the clouds and stuff. But yeah, so there's that. And then as far as the coding for the phycocyanin stuff, there is a paper out from Sarah Ekbarnajan-Nishelli and Lou McCaffrey. And so that came out, I think, a couple years ago. That's got a lot of the processing information in it, if you're familiar with it. So Google Earth Engine, you can use JavaScript or Python. You can do either in the interface. Yeah, so you can kind of look at how they determine algorithms and stuff in that paper.
No, I've actually read that paper a little bit for my
Nice.
my lit review. And, that's awesome. I didn't realize you could use Python for that, so that's really helpful. Thank you.
Well, we use JavaScript, but you can use either language within the Google Engine interface. It's like interchangeable.
Cool.
Because I don't know Python.
Yeah.
I'm also not very good at JavaScript, but I'm not good at any of it. But that's what Lou used. So I'm learning JavaScript now.
Yeah, I did into it for those reasons too. It's really cool.
Sure, yeah.
Awesome. Any other last questions or comments? Well, thank you guys for joining. Like I said, we'll follow up with this recording and the presentation. Thank you, Sarah and Lexi, for helping out. And I hope, and if you have any questions, like I said, reach out to us at the Finger Lakes Watershed Program or the HABs email. And happy, hopefully, NYHABS reporting season.
Hopefully it's quiet.
Yeah.